Dear students, today we are going to discuss the protoplast fusion, or you can say somatic cell hybridization. So normal plant cell contains cell membrane which is surrounded by the cell wall for much more rigidity, as you all know. And when we remove this cell wall, the cell is left with protoplast along with some other component. So now after removing the cell wall, we have protoplast in a hand, and we can fuse such 2 or 3 isolated protoplasts, but mostly we fuse 2 protoplasts generally for our convenience, which further developed into the new plant. And this is the concept of the uh, protoplast fusion. So there is a question that what is somatic? First of all, somatic means that any cell from, you can take any cell from the plant body, either you can take the cell from stem or leaf or root, except the reproductive organ. So that's why we called it somatic cell. That means you can take out the cell from any of these parts of the body of the plant and use this in this technology. Now with the help of the diagram, this with the help of this diagram, I would try to make you understand that what is the exactly protoplast fusion means. The purpose of somatic hybridization is one more important thing which you should have know, that what is the purpose of somatic cell hybridization. So the purpose of this somatic cell hybridization is to produce a genetically modified plant which have the better properties in them. So to understand it, I have just draw this diagram for your convenience, that this is the plant cell. Plant cell, as I told you, that plant cell had cell wall and cell membrane, of course, cytoplasm inside it. So we remove this cell wall and we left with the protoplast. Suppose we are taking two plants. One is plant 1 and this is the plant 2. And this plant 1 is disease resistant. That means no disease can occur in this plant, and this is the disease plant. That means you can say vulnerable plant. In future there's possibilities that any disease can this plant can catch any disease. So when we fuse both the protoplast of these two types of plant, and we get the a third kind of hybrid, that means it has the genetic content of both these two cells. Genetic content is now present into this third cell, which is called hybrid protoplast, and this hybrid protoplast is developed into the hybrid plant later on. And in this way you can produce a complete plant having two different genetic compositions. So this is the main this is the basic idea of the protoplast fusion. Now come to the next diagram. In this diagram I am going to explain you that what is exactly somatic cell hybridization, because in your examination so many times this question has been asked that describe the somatic cell hybridization, and this diagram is not given in your book. I have just drawn for you, so just see it carefully. In this process, first we are taking two cells. As you can see that these are the two cells intact with the cell wall and cell membrane, all these things. But later on we just break these cell wall with the help of enzyme that is called cellulase. Why the cellulase? Because we all know that the plant cell have the cell wall made up of cellulose. So we use the enzyme cellulase, and with the help of cellulase enzyme we break the cell wall. After breaking the cell wall, we know that there is a free protoplast, and we can fuse these free protoplast. So for fusion of these two protoplasts we need two things, or you can say we have two procedures, either which one you would like to follow. One is that called electric fusion. Electric fusion means with the help of a small current. That means we have to provide the small current to these two protoplasts and they both combine. Or another method is, if you don't want to use this electric current, you can go for the chemical fusion by adding some chemicals like PEG, that is polyethylene glycol, or some other chemicals. You can make the fuse two types of protoplast. So either method you can use, so according to which you fuse the protoplast. Now when protoplast fusion doesn't mean that only protoplast is going to fuse, it means that the content of their nucleus is also going to fuse, because this is the main motive of the production of the plant, that you want a hybrid variety which is better than their parents. So here in this diagram, fusion of protoplast occurs, and these both nucleus fuses and made a huge, or you can say giant nucleus. This giant nucleus is the has the content of both the two protoplasts, and this is called heterokaryon. Heterokaryon means hetero means different and karyon means nucleus. That means the source of nucleus is different in this hybrid protoplast. Now this hybrid protoplast is then placed into the nutrient medium in a test tube, and then as you all know that these cells will develop into the callus. Callus is the undifferentiated mass of the cell, and this callus is again developed into the small plantlets, and then we take out this is small plantlets and grow this plant on the soil, and this plant has the good properties of both these two protoplasts. That means suppose if you are taking these two protoplasms, one is giving you the good amount of crop, that means large quantity of crop, and this is the disease resistant protoplast, and we are fusing both these two protoplasts, so we get both the properties into this plant. That means this plant produce large number of grains as well as this is also disease resistant. So you mix the two best properties in a single plant. This is the main process of the somatic hybridization in this way. Now what are the you can say what are the various uses of this somatic cell hybridization, and what is the benefit of it. So this method is useful for the plants which are not fertile. Let's say that suppose plant is infertile, so that we can overcome this incompatibility of various plants. Now another thing is that this technique is a blessing for the farmers, as we can get the desired plant in a very less time as compared to the traditional hybridization, because you have studied that in traditional hybridization generally you take 4 to 8 generations. After then you can get that variety, improved variety which you want. But in this case that means somatic cell hybridization, within a few hours you can say you can produce this kind of protoplast, that means hybrid protoplast, and rest of the development may of course take a little time there, because you know that after fusing this protoplast it has to develop into the callus, then it develops into the plantlets and the complete plant. So that is the other story, but the basic purpose of the fusion of the protoplast which occurs into the 1 day 2 day, or you can say in hours only. So these are the two things. So first thing which you have just keep in your mind, that what is the purpose of this kind of hybridization. The purpose of this hybridization, or you can say the goal of the hybridization, is to mix the genetic content of the two best cells, or you can say desired cells which have the desired quality which you want to see in your future crop, so that you mix the genetic content, that means nucleus content, because cytoplasm content has been already mixed, that is no issue of it. But what is most important is to mixing of the chromosome, that means mixing of the DNAs, because we need it, or we produce is for the future generation, so we need the better varieties, you can say genetically better varieties in our progeny. That's why we just go for this process. That is all.